Hi, welcome to the screencast. This is going to be about macromolecules. We'll probably get through maybe two different macromolecule types in this first video. I'm going to break them up into shorter videos so that you're not sitting in front of the screen for as long. I don't recommend much more than 15 or 20 minutes of video watching at a stretch because your mind is certainly going to wander. So first up is macromolecules. By macro, we just mean very large. So because they're large, they're either used for building things or as energy. And just to sort of give you some sense of scale, this is a water molecule. I do not know if these things are to scale. I made them myself, but I tried to make these little bubbles about the same size as those bubbles. So if that is a water molecule, this is a protein. It would be a relatively small protein. So you can see that the size scale is vastly different, with water molecules being much, much smaller than these big macromolecules. So this is what we mean by macro, just very large. We are going to have four different types of macromolecules: carbohydrates, lipids, proteins, and nucleic acids, for each one, you want to know the name of the monomers or the parts that make up that macromolecule, and then also the polymers. And then you want to be able to identify pictures of the monomers and the polymers when you see them, and know the major function of each of the different macromolecules in the body. So those are the sorts of questions you're going to get on the exam. So we are starting with carbohydrates. They're called carbohydrates because they contain carbon, hydrogen, and oxygen. The monomers of carbohydrates are sugars or monosaccharides. The only one that I want you to know by name is this one down here, which is glucose. This is, as it says right here, the molecule your cells prefer to break down in order to make energy. So my shorthand for that is fuel for cells, but you can think of it as sort of like the gasoline that goes in the tank of your car. It is the preferred energy unit, if you will, for your cells. There are other monosaccharides, should you want to know them, like fructose, galactose. Here are the two sugars in your DNA and RNA, deoxy and ribose. Then there are some disaccharides. This is what table sugar looks like, sucrose. So it is one glucose and one fructose, I believe that is. And then there's lactose. That's the sugar that you find in milk. And maltose, I have no idea what that shows up in. Then the polysaccharide is the polymer of monosaccharides. The polysaccharide that we care about is glycogen. You do want to know that glycogen is a polymer of glucose. So each one of these little, stop sign looking things, is a glucose molecule, and this is how your body stores glucose, all linked together into this great big molecule called glycogen. And this is for short-term energy storage, as it says there. So the way I like to think of it is your daily energy budget. It's the extra glucose that your body's not using at the moment. This is what is stored in your liver and muscles to fuel your energy needs throughout the day. So you would then want to be able to identify something that looks like this as glycogen, and something that looks like this as glucose, and then know the functions of both glucose and glycogen. Next up then is nucleic acids. These are your DNA and your RNA. You don't need to be able to list everything that they are made out of. The building blocks of nucleic acids are nucleotides. So each nucleotide is composed of a sugar and a phosphate, and then this nitrogen-containing base. We call it a base because this NH2 group right here can absorb a hydrogen ion and become NH3. So anything that is going to lower the hydrogen ion concentration of a solution is called a base. So these we refer to usually as your nitrogenous bases or nitrogen-containing bases. There are 4 A, G, C, and T, and then U in your RNA, which we'll get to when we get to RNA. Sorry, I'm still getting used to using the new PowerPoints. So these are the bases: adenine, guanine, cytosine, and thymine. I'm sorry, this figure did not blow up well. I had some trouble getting the figures to work properly, so some of them got blurry when I enlarged them, and this is one of them. So whether you're talking about RNA or DNA, you start off by linking all the nucleotides together with the phosphate of one nucleotide linked to the sugar of the next. So they have what is called a sugar-phosphate backbone, where it goes sugar, phosphate, sugar, phosphate, sugar, phosphate, and then the nitrogen-containing bases sticking out into the middle. And you'll notice that some of the bases have 1 ring and some of them have 2 rings, so they're different sizes. That is going to become important when we talk about the structure of DNA more specifically. Let's get the next slide. So for DNA, the 4 bases are adenine, guanine, cytosine, and thymine, A, G, C, and T. The sugar is a deoxyribose sugar, so it's this sugar right here with, it's this oxygen right there that's missing. So with DNA, it is 2 strands held together by the sugar-phosphate backbones that get put together to make one big supermolecule. So again, you have sugar, phosphate, sugar, phosphate. So this is 1 strand. And then over here you have sugar, phosphate, sugar, phosphate, sugar, phosphate. So this is another strand here. And then the 2 strands are held together in the middle by these hydrogen bonds. So the different nitrogenous bases hydrogen bond with one another and hold the 2 strands together. So one of the things that I want you to notice is that there is specific pairing that goes on here, and we'll talk about this more when we do replication and transcription and translation, when we talk about how DNA actually works in chapter 3. But anywhere there's a G, across from it is going to be a C, and anywhere there's an A, across from it is going to be a T. So here's an A, there's a T. If this is a G, that's a C. And the way it works is that it's always one of the big bases with 2 rings across from one of the small bases with 1 ring. So 2, 1, 2, 1, 2, 1, 2 here, 1 here, 2 here, 1 there. What this does is ensure that the distance from one side of the backbone to the other is consistent the whole way down the strand. So you never have 2 nucleotides next to one another, which would cause, right, or I should say you never have 2 small nucleotides like that, 2 of the 1 ring bases across from one another, which would cause it to pinch in, or 2 big ones like that across from that, which would cause it, to bow out like that. So when you're looking at DNA, it's always uniform width in between the 2 sugar-phosphate backbones. The other thing that I want you to notice, whoops, I almost deleted the whole picture, is that the 2 strands are oriented in opposite directions. So if you look here at the bottom of this strand, it is the sugar of this nucleotide that is sticking out, and the phosphate that is associated with this nucleotide is here. And then on this side, you have the free phosphate that is exposed. And then if you look up here, it's just the opposite. So if this is the sugar end down here, this is the phosphate end up here, and then this side has just the sugar exposed. So there's directionality to the two strands, and they both run in the opposite direction. All of this is going to become more important when we talk about how DNA functions inside the cell. For now, we just want to notice these things. So this is how we usually show it. It's a little bit more stylized. So you have two strands that are held together by hydrogen bonds that end up forming this twisted shape called a helix. So because there's two strands twisted together, we call the final shape of DNA a double helix. The reason why DNA takes this final form is that this is a very energetically stable conformation or shape for the molecules to be in. And what this means is that the DNA molecule is mechanically stable long term. So it's not going to break. It's not going to chemically react with other things, which would change it. It's not going to get damaged as easily. It's just a very good way to keep the DNA intact, which you want to do because what your DNA does is code for proteins. So that means that there is information encoded in the sequence of nitrogenous bases, either on this strand or on that strand. And we're going to see later on that there are mechanisms in your cell that are used to read the DNA, the A, G, C, T, and turn that then into amino acids to make proteins. So then there is ribonucleic acid. So this is different from deoxyribonucleic acid in two important ways. One, the sugar is different, so it's a ribose sugar instead of a deoxyribose sugar. Actually, there's three things. Two is that instead of thymine, there is uracil. Where's uracil? There's uracil in your ribonucleic acid. And the third important thing is that RNA exists single-stranded. So you have one sugar phosphate backbone here. And in this picture here, it's the more stylized version of it where the sugar phosphate backbone is just this ribbon, and you can see the nitrogenous bases sticking out without a partner across from them. So it's just a single strand instead of double-stranded. Here they show it coiled up like a helix, but RNA forms all sorts of different weird shapes depending on what these nucleotides are going to stick to or what the bases are going to stick to. For us, the primary function of RNA is to serve as a, it's not said here. It is a copy of DNA. Oh, it is there. I'm sorry. I'm still getting used to reading my own slides. Disposable copy of the same information encoded in the DNA bases, which is shipped to the cytoplasm and then read by ribosomes to synthesize proteins. So you can think of DNA as being like the original blueprint for a building. And then RNA is a copy that gets sent to the construction site. So if somebody spills concrete or paint or coffee on it, you still have the original pristine blueprints at the architect's office somewhere or something like that. So that is DNA and RNA. So DNA, you want to remember, codes for proteins. It is the information needed by your cells to make proteins. And ribonucleic acid also codes for proteins, but it is disposable, whereas DNA is what's passed on from one cell to the next and one generation to the next to ensure continuity of populations throughout generations. Then we have ATP, which we need to talk about briefly. It is not part of DNA or RNA, but it is chemically associated with it, so we talk about it. So ATP is your adenosine nucleotide, only instead of with just one phosphate on it, there are 3 phosphates attached to it. So it is adenosine triphosphate. This is, as it says right here, the primary energy providing molecule for your cells. So the way you want to think about it is that a cell is maybe like your house, right? And you have lots of energy requiring things in your house. Some of them you plug into a wall. Some of them have batteries, right? You have rechargeable batteries. You have D cell batteries, A cell batteries, AAA batteries, the little flat batteries that you put in your watch. That's a lot to keep track of. So what your cells have decided to do, or what has happened throughout evolution, is that there is just one battery that fits all of the energy requiring machinery inside of the cell. So when you eat carbs or proteins or fats, your body is going to convert all of those into ultimately CO2 and water. And then the energy used from the breakdown of carbs, proteins, and fats is used to make ATP, and then ATP is what fuels everything. So it's sort of like you can use coal or wind or solar to make electricity, but electricity is what makes a lot of the stuff go in your house. Same with your body. You can use carbs or fats or proteins to make ATP, but ATP is the primary energy source for any single process inside of your body. So we are going to stop there at 17 minutes. Nice bite-sized pieces of information, and we will pick up in the next screencast with lipids.